non-essential and essential transcription factors in MTB and investigating important host pathways that can be used for host-directed therapy. Dr. Vinay Kumar has been bestowed with many prestigious awards such as National Bioscience Award for Career Development, one of the highest Indian science awards by the Department of Biotechnology, Government of India. He also holds the prestigious J.C. Bose Fellowship, and Dr. Vinay Kumar is an elected fellow of the Indian Academy of Sciences, Indian National Science Academy and National Academy of Sciences, India. With this brief introduction, I would now request Dr. Vinay K. Nandakuri sir onto the dais to deliver his talk. Please join me in welcoming sir.
Morning, everybody. Thank you for the invitation and the opportunity to talk about something very different from what I do in my own laboratory. For last 3 years, we faced a major pandemic, and we have done quite a good job of both vaccinations as well as diagnostics and many other aspects of controlling or at least making sure that reasons are, it's done well enough during the pandemic. But we wonder, is it the last one? I'm not sure. I don't think that's going to be the case. Was it the first one? Even that is not the case. So what I'm going to do today is briefly talk about a basis, a molecular basis of why we get it, and what is the history of pandemics, and how viruses or bacteria actually evolve. That's the brief outline of today's talk, and that's what I was saying. It has nothing to do with what I work on, which is on Mycobacterium tuberculosis. So this is how all our body has almost 10 to the power of 13 cells. And most of them, all of them look very much like this. They have DNA, in the nucleus, and then you have protein factories outside. You have mitochondria where energy is produced, all these things. And all these cells in our body are interconnected. And we have various organs, which including very strong immune system. But it's not that we are not under attack. It's almost like our body is always constantly under attack by other things. So what happens in our body is that we have DNA, which is the genetic material, then gets made into an RNA, which is actually the copy of it, except that it is there for a shorter while. This RNA becomes the template for producing the protein. And RNA is also involved in many other functionalities itself. This is what we call in science as central dogma, and this was something that came up 70, 80 years ago. Over a period of time, people realized RNA can be made back into DNA. DNA, of course, replicates itself, and so there are other exceptions that started coming. And I was saying that our body we are actually always face attacks from outside things. What are the kind of infections one can get? I'm sure all of you know it. Bacterial infections, staphylococcus, sometimes you get a throat infection, you have a severe throat pain, and you get a chest infection, pneumonia. These are all bacterial infections. And then you have fungal infections. Fungal infections are basically, sometimes you get dark skin patches on people's skin, and they're generally caused by fungus. And they can even happen in the internal organs. Parasitic infections, malaria is a very good example of a parasitic infection, where people end up getting bitten by a mosquito. Mosquito carries the Plasmodium falciparum. You end up getting malaria. You end up getting pretty severe fever. Prion disease, which has nothing to do with any of these things, but nevertheless it's an infection. And you have bacterial, and in between we have viral infection. So viral infections became quite popular in the sense that last 2 years, because of the fact that we have faced pandemic, everybody knows, yeah, we have COVID, right? Bacterial infections are, tuberculosis is a terrific example of bacterial infection, and some of the others which I will talk about. There are so many bacteria that can infect us. You can see this. Okay. So many bacteria that can infect us. And so it's not like we are free of bacteria in our own body. You may not know, or you may know already, that our body, let's say, has 10 to the power of 13 cells. We have 1.3 into 10 to the power of 13 bacteria in our body. And such bacteria are called microbiome. And these bacteria help us in many facets, including producing metabolites that are important for our body to function effectively. You have microbiota in your mouth, you have microbiota in your gut, and you have microbiota even on your skin, and you have microbiota in almost quite a lot of open parts of our body. And these actually have a pretty much serious set of influences on this. Now, viral infections, there are around 200 of them too. Viral infections, viruses are a bit different. Bacteria, in one hand, can survive outside, and it can replicate in a media. If you take bacteria out and you put it into some kind of a selective media, it actually can replicate, and you can actually make it produce another bacteria and eventually propagate it. Viruses cannot be propagated by themselves. Viruses only can be propagated if they have a host. So what kind of a host are we talking about? Host can be anything. If the host is a bat, it's at that point of time it is specific to the bat. If it jumps from bat to humans, then humans become the host. If it goes from us to a cow, cow becomes the host. So, so are we all same? To an extent, yes, but not completely. So the jump from one place to the other is basically is one of the reasons why all of a sudden you end up seeing new emergence of new viruses. Why does it, why does this kind of thing happens? It happens. That is the way nature is. Viruses that have a particular host, ultimately one find they jump the host, and these kind of things continuously happens from animals to humans, humans back into animals. This cycle continues. The process of looking at all these things is nowadays called One Health, right? So now when it comes to viruses, they cannot be propagated outside, but there are ways to propagate a virus. They are generally cell culture models which can be used for propagating the virus. So like anything else, anything that can be propagated has to have a genetic material. The genetic material in viruses can be either DNA or RNA. If it is a DNA virus, then it is called DNA. DNA is the genetic material. It's called DNA is the genetic material. It's called DNA virus. If RNA is the genetic material, it will be called RNA virus. If you have a DNA virus, it can have an envelope. It need not have an envelope. It can have single-stranded DNA. It can have double-stranded DNA. So all these things put together, you end up getting lots of varieties of viruses. For example, double-stranded DNA has a virus called adenovirus. There are examples of each of them, and there are multiple viruses of each kind that always infect us on a regular basis. And then you have RNA viruses. And then you have RNA viruses. Again, the genetic material can be double-stranded. Genetic material can be single-stranded. Genetic material can be negative sense RNA, positive sense RNA, depending on what becomes a template eventually. So all these things put together, they come under the concept of RNA viruses. Good example of an RNA virus everybody knows is HIV. HIV is a virus that causes AIDS. But these are one of the primary examples that were there. And then comes the major terms which are routinely being used for the last 2 years. Endemic, epidemic, pandemic. What is endemic? Anybody knows? One example of an endemic. For example, in a particular region of a country, of a particular region, you always have some infection coming, but that is limited to that area. A good example will be, let us say, malaria is endemic to, say, one part of, like, in this particular part of the world. There's a way of saying that in that particular, Ebola was endemic. At some point of time, it came up in Africa, but it stayed in a particular region, right? That kind of a thing. Endemic is more, again, also not, Ebola is not a good example. Something that comes very frequently, but it comes in one part of the world, not all over the world. That's endemic. And then you have epidemic. Okay? What is an epidemic? It's an outbreak. It's an outbreak of any kind. Ebola is a good example of an epidemic, actually. But it is not all over the world. It's come at a particular point of time. A lot of people are infected in that particular part of the world, but it is not in every part of the world at the same time. Then it is called an epidemic, right? So epidemics are many kinds. You can have local epidemics. SARS-CoV was not this one, but the previous one. H1N1 was at some point of time an epidemic. And then you have pandemic. It breaks out across the world. It spreads everywhere in the world. Then it's called pandemic. Here is the thing. Endemic is something that is present in certain regions of the world, and it comes at certain times again and again. And then you have epidemic is a breakout infection that happens. When that breakout spreads to the rest of the world, it becomes pandemic. So, people at some point of time were not moving as much, right? It's all kind of, the pandemics is basically an infection that has happened in a particular area but starts spreading to other parts of the world. It can only spread when people travel. So the first pandemic started happening in only after, when this kind of a movement across the world has started, because people started moving from one place to the other by ships, right? The first known pandemic is basically called Plague of Justinian, and this has happened in 541 after death. I mean, this is all in the like 2000. It was 541, and it killed quite a lot of people, 3 to 5 crores. And then came Black Death that happened in 13th century, 1334 to 1353. It killed almost 20 crore people. In fact, it says that it's kind of killed 1/3 of Europe's population at that time. And then you have world smallpox, which came a bit later in 15th century. It's also killed quite a good number of people. And plague, once again, came back in 1885, killing quite a lot of people. Spanish flu is what more or less everybody recently remembers, happened around 100 years ago, and that happened during or immediately or during and aftermath of First World War. And it was a perfect recipe, because the First World War was actually going on. People were in very close quarters, and a lot of soldiers were in not so hygienic conditions. Given all these things, it has killed quite a lot of people, 10 crore people almost it killed. And then we may not even remember, but HIV/AIDS was at some point of time was a pandemic. A lot of people were dying across the world, and that also killed almost 5 crore people across the world. And the most recent one, that is our memory doesn't go that far, is what we talk about is SARS-CoV-2. So the Plague of Justinian, that is basically named after the Emperor Justinian, because it was his period. And what it is, caused by a bacteria called Yersinia pestis, and that one of the carriers of these things are basically rats and some of those small flea kind of a thing. So what it does is that it disables the immune system by injecting toxins into your defense cells. That's what it does. And it spreads through rats, mice, chipmunks, prairie dogs, rabbits, and squirrels. so very easy to spread. It causes bubonic plague, pneumonic plague, septicemic plague. Depending on the symptoms you get, they have different names. But essentially, you end up getting some pretty much serious amount of this plague actually leads to death. You may not, I mean, many of you may not remember, there was a plague breakout in India, I think in about 20 years ago or so, in Surat area. So, generally it doesn't come anymore because our hygienic conditions are way better today than what it used to be way back in those times. But once it comes, relatively easier to stop the spread also. Smallpox is something pretty severe, and it has killed a lot of people at some point of time. So this was actually the first time a vaccine was kind of invented by Edward Jenner. What he has done is that they had a cowpox, which is essentially happening in cows. What he has done is he's taken something from one of these things and put it into the people who are not infected, and they started getting protection. Or basically it came from the concept that people who were working with cows were not getting severe symptoms of smallpox. That's the story, basically. That's what he has observed, that whoever is working with cows, like, people, maids who basically pull out the milk and all this, those people were not as exposed to smallpox, or were not getting as severe symptoms with smallpox compared to the others, which essentially meant that there is some protection is being given by the exposure to something the cow has. That is generally cowpox, which is a bit different from smallpox, which is adapted to us as the host. So based on that, he actually came up with this vaccine concept, and this is the first vaccine to ever be used. What is a vaccine? It's like somebody, you have an open house. Let's say we all have many places in quite a lot of time. Nowadays it's not very usual that people close the doors. But at some point of time when I was growing up, in many villages and many places, hardly doors were closed. In fact, they were kind of open, right? People would stream in and stream out. Now consider our cell as something like that, our body as something like that. But people are coming and going, many of them are friendly, you really are quite all right with it. But you may have some of them who will come and steal something from your house. So, however, if you actually recognize that person, next time around you will be able to stop it. That's essentially the concept of how immune system works. Immune system is basically making your body aware that whoever is walking in is actually, whoever is coming into your body is actually the enemy. So when you are somehow basically telling the immune system, here is a strain, if it comes again, be careful, and I should have an immune response to kill it. And that is essentially vaccine. Now, vaccines can be given in multiple ways. We'll come to that. And influenza virus, that is basically what has caused the flu. This is essentially, influenza virus is not new. Influenza virus has been around for a while too. But it so happened during the World War I time, influenza virus became something more virulent, and the infection started breaking out originally in the military camps and eventually spread across the world. And influenza virus, like COVID, also came in multiple waves. The first wave caused death, but it subsided. But somewhere along some host, it persisted, it became better, and came back to give a huge second wave, killing so many people, including in India, in Bombay, and in many other cities across India. A lot of people died of influenza virus. And eventually there was a smaller third wave, and the immunity developed, and it's not like it went away, right? It kept on coming. Every now and then you will have a small outbreak of an influenza infection. Last one you may have heard of is the H1N1 in 2009. So around that time, all of a sudden there was a huge talk about influenza virus and H1N1 infections. And a lot of hospitals had places where people were secluded if they were to have H1N1 infection. So how do you stop a virus of influenza kind? There are two ways to treat anything that's called an infection. One way is obviously to make your body aware, which is called vaccine. The other aspect is treat once the infection happens. So there is a particular drug that was used very extensively and even now used. It's called Tamiflu. So Tamiflu is a drug that acts on basically stops basically viruses from replicating more. So even if you were to have an infection, if you can recognize and catch the infection soon enough, you can go on Tamiflu and get cured much faster. But there was a window. The window is only first 48 hours in which Tamiflu works. After that, it doesn't work any more effectively, right? Then came the last one that we know of, is called human immunodeficiency virus. I mean, do you know what is the size of our genome? How many of you know what is the size of our genome? 10 to the power of 9 bases, roughly, right? 10 to the power of 9 bases. Do you know what is the size of HIV? The virus that causes HIV? 10,000 bases, roughly. I'm not giving you the exact numbers. 10,000 bases. So 10,000 is essentially 10 to the power of 4. We are 10 to the power of 9. Something with 10,000 bases can create havoc in our body, which has 10 to the power of 9 bases in each cell, and it really creates havoc quite a lot, right? And these 10,000 bases also have the tremendous ability to mutate and adapt so that they cannot even be eliminated that easily. They persist in our body. The biggest problem with H1, the HIV, is it persists, right? It persists in our body, and there is a therapy. The therapy is not a single drug. If you end up giving a single drug to either a virus or a bacterial infection, what you would end up getting is you would end up getting resistance over a period of time. So what I'm talking about is HIV infection, which is essentially immunodeficiency. It does not kill you. In HIV's infection, people don't die because of HIV. They die because of the secondary infections. HIV essentially compromises your immunity beyond repair. And then other infections come and eliminate you. Other infections cause the eventual death. Means your body becomes defenseless, right? Final but not the least is SARS-CoV-2. SARS-CoV-2, this is a old statistics, is nothing new. But even if it has not gone up, it's gone up to 70 lakhs or so. SARS-CoV-2 is basically, we were second in number. Now China also has a lot of new cases in the recent past. SARS-CoV-2 is caused by a virus, which is, the COVID is caused by a virus called SARS-CoV-2. You can see this. So this virus actually has a genetic material, not 10,000. It is considered a reasonably larger virus with 30,000 bases. Okay, it's a single-stranded RNA virus. It has 30,000 bases. It has 30 plus viral proteins, and like any virus, it has certain way to enter your body. It is through a surface protein called spike. You may have heard of this term a lot. Mutations, spike, and variants. These are some of the terms. RT-PCR. All these are the terms which school students to a commoner to anybody has heard in the last 2 years. If I would have said any of these terms 3 years ago in a general public, they would have looked at me like, what are you talking about? Now at this point of time, they will say, yeah, they will give you a pretty knowledgeable nod, right? Yeah, these are the things we know. And it's all because of what they have seen in the last 3 years. So 30,000 bases, it has obviously virus, it has even enzymes to replicate itself. Okay, RNA-dependent RNA polymerase is an enzyme that is required to replicate itself, and it has spike to make an entry, and it has many other things that are needed for it to be a very efficient package, come back and get out, right? It looks like this. The other term that most of you overheard over a period of time is called variants, right? A variant essentially is, again going back to the example of a thief, you have recognized the face of the thief, so you are careful if the same thief comes back. Now let us say the thief comes back with a mustache, right? You may not be able to immediately pick it, right? You may be vigilant, you may be suspicious, but you are not able to pick. So now again there will be some level of this thing. But within a little bit of time, you realize that, oh, this is the same thief except come with a mustache. Means severity will be a bit different because you have multiple layers of defense in your body. You have, when any kind of a pathogen enters our body, we have innate immune response that comes into play. That involves such as, macrophages, dendritic cells and other things. They take up the pathogen that they see, or that they take up something that is new. And then they present it, and you have adaptive immune response that comes into play, which is B cells and T cells. B cells produce antibodies. That's why called antibody therapy. We talk about it a lot. And the T cells are essentially, they can kill the viruses themselves. So these, even if your innate immunity fails to recognize the enemy, at some point of time, your adaptive immune response, or T cells or some of the other T cell mediated immunity or some other things come into play, and you might eventually kill the virus, which is which is slightly different from what was originally the case. So this is, why does it happen? People keep on asking, why are we getting new viruses? Why do we, how many variants are going to come? Are we going to get new variants going forward? It's essentially survival strategy, right? We have produced a vaccine to kill it. We have some kind of medicines now that are coming up, which will kill the virus. Now, virus and us, it's like a cat and mouse game. They also have to survive. For that, what they do is they constantly evolve, or they constantly make mutation. Now, how do they know what mutation is important or good enough for them? You make 10 or you make 100, one will be good, 99 may not be good. So that one gets selective advantage over a period of time. Eventually, you end up getting a mutant virus. So there are two levels of terms that are used. One is variant of concern, and there is variant of interest. So variant of interest is, it has some mutations, but those mutations may not be persistent in a larger population in the infection. In which case, there's a variant of interest. We are watching it. We'll see whether it's going to. Variant of concern is basically it does cause a bunch of infections. And then you have variants, you have subvariants. These things keep on happening. For example, here are some of the variants of concern that you may have heard of: alpha, beta, gamma, delta, omicron. These are the variants of concern that kind of got developed over a period of time. And then you have So then you have variants. Once a variant of interest becomes variant of concern when it becomes too many infections. So essentially, India had 3 waves. The first wave was mostly caused by the original variant called Wuhan variant, and then the second wave is caused by Delta variant, which you see here. And the final wave in India, which is after that also, we do have Omicron infections going on, is caused by the last strain Omicron. So what has changed is that each variant evolved. There were certain infections, certain mutations that got accumulated in the spike protein. And Omicron actually incorporated so much more than anything else. Here are the various mutations that are seen in Omicron. So where are we as far as the pandemic is concerned today? In the last 1 year, that's since Omicron has arrived, a little more than a year, 1 year, 2 months, there are approximately 300 odd Omicron strains have come across the world. India alone has faced more than 250 variants of Omicron. Some of them obviously have caused more infections than others, but there are variants of Omicron that are still going around in India. And if that is the case, why pandemic is not exactly a pandemic anymore? Answer is not very complicated. We all, 80% to 90% of the population in India is vaccinated with a virus, vaccinated against this virus. The vaccine that we have taken is original virus, Wuhan variant, but nevertheless there are 2 levels of immunity: innate, which produces And then you have adaptive, which are again two arms, a production of antibodies, and you have T cells. So given all these things, we have a immunity coming from vaccination. In addition, over the last 3 years, we have been infected extensively by various forms of the same virus, sometimes multiple times. And all these things essentially means that we have been told again and again, here is a virus, you should recognize next time around they come. Means we have developed certain level of immune response. Does it again infect you? It comes with a beard now, maybe. In the sense, Omicron may become something else. You may end up getting one more variant. We may end up getting infections. We hope one arm of our immunity or other would be able to help us in not making the disease more severe. So this is actually how we are actually going to take care of, why we don't have the levels of infection what we had. I personally believe virus is still going around. Many of us are getting certain level of symptoms, but now the symptoms are not severe enough for us to get alarmed, maybe, and that is why the hospitalizations are down. When the hospitalizations are down, you realistically are not going to be heavily alarmed by the infection that are going around. It's not this, you get some other cold. It is not cold, you get flu. But this is how it is. It's a war that is going on essentially between us and viruses and bacteria. As long as we are, our immune responses are good, we will be in a better shape. And as long as the modern medicine has developed well enough, and there are ways to make sure that people, when they're at the weakest moment, are taken care of, most probably the survival rate is likely to be higher, and which is the reason why the death rate during the third pandemic, the recent pandemic, is not comparable to the death that is observed during Spanish flu. So, why? World has changed around us. In the sense, when Spanish flu came, people did not have any way to find out what is the sequence of a Spanish flu. Now we can. These are called surveillance strategies. Zero surveillance allows us to find out who's infected, who's not infected, what is the level of antibody they have in their blood. Air surveillance allows us to figure out if it is traveling through air and how the infection is spreading. And sewage surveillance gives us an idea about the extent of infection in a larger population, because whenever anybody gets infected by a virus, they also shed virus in their feces. And genome sequencing allows us to figure out what are the sequences and what kind of mutations are giving it an advantage/disadvantage and all those things. These are combined with laboratory studies. And that is one aspect. You will be able to combat anything effectively if you know you are actually infected. That's where diagnostics come into play. There has been serious advancement in terms of diagnostics, and the cost of diagnostic for a pathogen like SARS-CoV-2 has become much better with time, and in fact, it is now costs much less to get a diagnostic done for COVID. Vaccine obviously helps, because then if you're vaccinating a population, you're making sure that the population actually does not show very severe symptoms, even if they get infected again. And the last one, but not the least, is treatment. This option is a continuous exploration that goes on. We have to develop small molecules that will inhibit certain of these virus enzymes, and eventually some of these small molecules will make it to the market, and these are the ones which will be used as drugs. And so combination of all these things, we can actually control a pandemic better. But this is not the first pandemic, nor would it be the last pandemic. The frequency of the pandemic is determined by many things. The transmission from animals to human is called zoonosis. Back from humans to animals is called reverse zoonosis. And the way our environmental temperatures, water bodies, and all these things, the bacteria, viruses that are propagated in and around, everything keeps on changing. It's a dynamic environment. We are part of this dynamic environment. We are not determining completely the dynamic environment. We are part of it. When it comes to going forward, where the world is moving, is to look at all of these things together. It is not anymore a human health, not simply animal health, not bacterial, not an environmental issue. It is a totally holistic view of whole thing. And simply put, it is called One Health. That is a buzzword nowadays. The problems going forward are the fact that there are lots of bacterial infections which are drug resistance, called antimicrobial resistance. And why is that a problem? There are so much drugs that are consumed by us even when it is not needed, that these bacteria can actually develop resistance over a period of time. If you are having a drug resistant bacteria, then you do not have a treatment option available to you. Again, it will not be a problem for a younger population. Relatively, they will be able to manage it. Our life expectancy has gone up. People are much older now relative to what they were when there's an older population of people across the world has gone up. In India also it has gone up, which essentially means that you are going to face more and more of other issues, drug-resistant bacteria becoming an issue, pandemics becoming an issue, or some of these things. These things will continue to happen. So this is the reality. And the other thing that has happened over a period of time is, if you would have told me, if you were an engineer, what is your business with biology? That's not anymore the case. Engineers are the ones who develop everything that we need for doing sequencings or analysis of proteins. Every biological instrument in the world is developed by an engineer. So engineers are integral part of addressing biology. The 19th century, the last century, is essentially the century where chemistry and physics thrived. And this century would be the century where biology would thrive, not alone. Will be central. You will have chemistry coming in the form of drugs. You have physics coming in the form of understanding how biology is working, even the cell membrane moves, how the dynamics of actin cytoskeleton network work, and many other things. And then you have engineering coming into the context of biology, in the context of new machines, highly intricate machines, next generation sequencing. And then we have huge amount of data that is generated because of that. So computational biology is integral part of biology. There is huge amount of big data that is going to come, and this big data, this is where the future is. So biology is now an integrative biology, and this is where we are going to go. So do not think that we are living in isolation. The world is going towards more of a collaborative way of working together, collaborative in multiple levels, looking at animal health, looking at human health, looking at environmental, everything in parallel, and also looking into big data, to everything, to solve some of these questions. With this, I would let you think more about some of the aspects of this. As I was saying, my personal research is on a bacteria called Mycobacterium tuberculosis, and it has only 4,000 genes, but it is present in Egyptian mummies to us, right? Nothing is gone, and even today it is a problem. It kills approximately 15 lakh people every year across the world, continues to kill people. So we continue to work on Mycobacterium tuberculosis, and viruses, they are going to be around. There are people who are going to continue working on it. Big data is where it is going to be in future. Thank you.
Thank you, sir. The session is now open for questions. We can take few questions from the participants if you have any.
Can't hear you very well. Not yet, at least.
Hello, hello.
Yeah.
Due to the accidental viruses of diseases in future generation, how can we expansion of human life?
Viruses and bacteria have been around as long as we've been around. In one
Okay.
sense, it's not new. It is not something new. I mean, you're saying some kind of an engineering and that is leading to this. Or is it simply the existing set of viruses and zoonotic transmissions that are going to happen from animals to humans? Whatever it is, it's not new, except that we have much better ways to deal with the current challenges that are coming. So if with respect to the human health, we have better handled today than what we had 20 years ago to deal with any of these major infections. Okay, and our medical facilities have advanced well enough that the survival rates are better even when people fall sick, right? And we have better, we were able to produce a vaccine within, I mean, world is able to produce a vaccine within 1 year of the infection, which was unprecedented. So better handle. In fact, we are in a better shape at this point of time going forward than what we were, say, 30 years ago or 50 years ago.
Good morning, sir.
She's saying something, let her finish. Yeah.
You said that your work is done on mycobacteria. No, we have a. Yeah, I start my. Good morning, sir. Since you have said that your work is done mostly on Mycobacterium tuberculosis, now we have many strains of Mycobacterium tuberculosis which are drug resistant. Even the combined drug therapy isn't working on them. And since then we are seeing little strains of Mycobacterium also. So what's the progression of work that is done in this particular field?
There are, this can be answered twofold. You have to understand, in Mycobacterium tuberculosis, whoever has TB, you may have seen people with TB.
Yes.
Therapy is 6 months. Combination of drugs. And generally what happens is when you start taking these drugs, in about 2 months' time, you start feeling pretty good. Means you're not anymore having the symptoms that actually made you feel terrible. Over a period of time, between 3 or 4 months or so, people tend to slowly go away from the drug. And that sometimes causes what we call as compliance issues. So the compliance issues actually result sometimes in drug resistant strains. And again, drug resistance happens because of mutations in the bacteria. And Mycobacterium tuberculosis has a size of 4.2 into 10 to the power of 6 size. And these bacteria, there are some spontaneous mutations that happen. Some of these spontaneous mutations either change the ability of the drug to act on its target, or they change the drug itself, or change the intracellular concentration of the drug inside the cell. Any of these things can change the effectiveness of the drug, and it is the reality. So there are, how do you take care of it? One, better knowledge dissipation to stop misuse of the drugs. And make sure that patients and others' compliance is better. Second, a constant work towards evolving and identifying novel drugs which are not in use currently. In fact, if you have a multi-drug resistant mycobacterial infection, it is treated by 2 other new drugs. They are only used when everything else fails, called bedaquiline and delamanide.
Thank you, sir.
Good morning, sir. Sir, why we are failed in developing vaccine? Why we are failed in developing vaccine against HIV/AIDS?
Okay, HIV/AIDS continues to evolve and it evolves very fast. It accumulates mutations very fast. So now let's assume that I take one of the HIV proteins and we inject, we basically put it in a template and let your body know that this is HIV. But the HIV that is coming in will be so different at a later point of time, so its evolution is faster. But the advantage with HIV is HIV doesn't live very long outside in the aerosol or on the surfaces. So HIV has a shorter time to live outside, so it didn't spread through that. It mostly spread to human contact. It can be anything, injected with HIV, or it can be whichever way of the human contact. That's how it spread. So vaccine could not be developed because of that. So any vaccine is only successful if you have one, variations don't come very fast. Second, your adaptive immunity, which is what's supposed to give you the protection, gives protection against the eventual infection. But if the vaccine is not able to recognize, or if your adaptive immunity and all these things are not able to recognize a strain that's coming in, then that will not work. So some viruses can be effectively treated with vaccine. In some viruses, we go for therapy. In case of HIV, it is therapy.
Thank you, sir.
Thank you, sir. It's very informative session, sir. And my question is, a few years back we heard about Nipah virus. So Nipah virus and coronavirus both are means almost similar, I think.
No, I don't think so. Even with respect to even coronavirus, a variant to variant, the differences are plenty. So, similarity is in the largest context, but let me tell you that before I answer this question, disclaimer, I don't know much about Nipah virus. I've not looked at it. In the sense, I've not read much about it as I should have, but I did not. But I believe that even within just now, as the gentleman asked a question about HIV is on 10,000 bases. We are still not able to develop as much of a vaccine against HIV because the variant that is coming is a bit different from what's already there. When you say something is similar, even a 90% similarity is not similar in the context of biology. We are very similar to monkeys in terms of our genome, but we are not similar, right? So there is a lot of similarity, but there are those differences that matter. So I have no idea in terms of the specifics because I have to look into genome sequence of Nipah virus and then you look into the genome sequence of SARS-CoV-2, compare and see what is the extent of similarity, where the similarity is, what are the genes that are similar, then I will be able to answer that question better. In the absence of that, I think any answer that I give will not be appropriate.
Okay, sir. Thank you.
It's like this. What's expiry essentially means that it is not good enough for treatment of people after that. If you do not dispose things properly and if it gets into our water streams, essentially we have antibiotics at a lower concentration that's floating around. That's what you're trying to tell me, correct? Not mycobacteria alone, it's not disposal alone. There are many reasons that cause drug resistance as far as bacterial infections are concerned. For example, in poultry farms across the world, a lot of antibiotics are used. Okay, and so which essentially means there will be things coming from there into humans, and if it's used irrationally, eventually you can have resistance because once a strain becomes resistant in some form, it will move around. So there are many reasons for it. We at least should make sure that antibiotics that are, in India, you can walk into a pharmacy and buy antibiotics. Realistically, don't need prescription, right? At some point of time, there has to be a proper enough regulations that antibiotics are only purchasable if they have a proper prescription and nothing else. So tighter regulation, making sure that it is not used in every place and it is actually disposed of properly, and it is also people are more compliant about antibiotics will improve our decrease the chances of resistance. Till all these things are in place, there will be evolution of resistance.
Good morning, sir. Good morning, sir.
Still can't see.
Sir,
Okay, there.
sir, why we can't find as early as possible a new virus because we have a more technology comparing to olden days?
You can identify a virus, not at all difficult. You can actually identify a new virus quite comfortably if you actually can isolate the virus, DNA, RNA, whatever it is, and perform genome sequence. Not only that we will also identify the sequence. But the point remains, everybody who goes to a hospital, if we are going to take their virus or whatever infection they have and get a genome sequence done, to genomic sequencing done to identify, that's a costly way to identify viruses. So whenever there is routine infections or whatever is not a major concern, people actually try to treat it with the existing knowledge and empirical way. Okay, when a particular place has too many
On behalf of T-Spheres and TT-Vres, we would like to extend our heartfelt thanks to Dr. Vinay K. Nandakuri sir for delivering a key lecture which is very important, and for his gracious presence today for the international conference BioMe 23.
Hello. On this wonderful morning, I take the honor of welcoming our eminent guests, distinguished speakers, delegates, researchers, academicians, and participants for your gracious presence to the Valedictory ceremony of BioMe 23, an international conference on advances in biology and medicine, organized by Telangana Social and Tribal Welfare Residential Educational Institution Society in association with Telangana Life Sciences, Federation of Asian Biotech Associations, Baba Academy, and Heartfulness Education Trust. We are immensely grateful to the government of Telangana for its generous support. This day is really remarkable with its impending impact on T-Spheres and T-Tveries. Just as the warmth and effect of the sun casted during day and can still be felt over the arena even at the dusk, I am still delving in the realms of facts put forth by our eminent guests in the past 2 days. Though the sessions have ended and the conference is moving towards its valediction, the valuable knowledge shared here will remain with us, guiding us towards a better future. Be it the malignancy of cancer with its looming cast on blooming lives, or the various means by which our pharma and therapeutic fields have developed over the years with possibilities of miracles. The facts we collected here will definitely lead us to be an improvised learner striving to do something further miracles.
I sincerely thank all our eminent speakers for enlightening us through their talks in the various areas of biology and medicine. We are bestowed with the presence of our esteemed guests with us. With immense pride, I take this opportunity to welcome our distinguished dignitaries who have gathered here in this valedictory. With immense pleasure, I now welcome Dr. Rakesh Mishra sir onto the stage to share his talk. We welcome you, sir. I welcome my colleague P. Sindura to introduce the speaker. Do that.
I consider it a great honor to introduce our distinguished speaker, Dr. Rakesh Mishra. Dr. Rakesh Mishra is currently the director of the Tata Institute for Genetics and Society, Bangalore. He is also Distinguished Emeritus Professor, Academy of Scientific and Innovative Research, and J.C. Bose National Fellow at CCMB. Under his leadership, the Atal Incubation Center was established at CCMB, which provides life science entrepreneurs the infrastructure and mentorship needed to encourage innovation. CCMB has also played a key role in genomic surveillance of the SARS-CoV-2 virus in India during the COVID-19 pandemic and made major contributions in diagnostics, drug screening, environmental surveillance, and capacity building in various efforts against the pandemic. His research interests are comparative genomics of non-coding DNA in the context of evolution of complexity, genetic basis of anterior-posterior body axis formation in animals, and role of epigenetic regulation in development. He also has keen interest in environmental and genomic surveillance of infectious diseases and application of cutting-edge science and technology in addressing societal problems. Dr. Mishra has published around 190 scientific papers and is a recipient of number of recognitions, including being elected Fellow of the National Academies, Indian National Science Academy, Indian Academy of Sciences, and National Academy of Sciences, India. We are indeed blessed to listen to your talk, sir.
Hello. Yeah, I think this is better. Yeah. So first of all, thank you all for being here to listen to what I have to say, and special thanks to organizers for inviting me to this wonderful place. It's always pleasure to come in this the effort to convey whatever some message we have from the science. And it's an amazing campus here. I came here first time. It's a really wonderful place to be. You already feel enlightened being in the nature and also with modern facility. So thank you again for all these arrangements. And what I'm going to tell, I just now understood that Vinay, the director of CCMB, has already spoken to you about pandemic and what kind of impact it has. I'm going to, so you are now very well introduced, so I can save some time and in the beginning go a little bit fast and tell you how we have learned from pandemic to take care in future such events and also take care of many of health issues which are quite prevalent and bothersome even without the pandemic. So we will go a little bit more into details of genome and environmental surveillance in what we call as One Health, which is health of everything. Human health is not isolated from the nature's health, animal health, wildlife health. These are all related. And if one component of the planet is not healthy, we will come into the same circle and will suffer. So that's why this idea of One Health is now worldwide. Government of India has made a special initiatives in many cities is starting to focus on one health and integrate other activities in that context. So just I will tell you that when we are not prepared or the world has become so small that any disease can spread so fast from one place to other, this is the maybe few weeks old, the, I think okay, it's very big. Yeah, okay. So already so many deaths have already happened in the world. India reported death is 5.5, which probably will be different if you consider all other published things. It may go many fold up. And as you are seeing currently, China is going through the massive something is all right. Okay. So in addition to this death, what we have noticed and the sufferings, the the economic disruption that COVID has caused over the world is now well over $16 trillion, and the psychological impact, break in education that it has for 2 years for the younger population, we are yet to even estimate that what impact it has. So these are the things which can create such a huge disruption in our regular activities that it cannot be ignored. And pandemic has also changed because of this high impact. Pandemic has changed the healthcare approach. It has catalyzed the research. The way we have made progress in past 2-3 years is just amazing. From a new virus to vaccine within 1 year, which has saved millions and millions of lives, even we were not able to imagine that we will be able to come up with a vaccine option for this disease. Vaccines usually take 10, 12 years to come, but it was within 1 year vaccine was given to people. And testing and surveillance at unprecedented level. At one time, India was doing 1 million tests per day, and these tests are very sophisticated that you make RNA, then you isolate from the patients, then you do a very sophisticated PCR and so on. And all this has helped in understanding the disease, how the virus is evolving, how it is escaping our immune system, and we can even track the virus from where it has spread because of these surveillance methods. So what is the surveillance method? Why we talk about surveillance so much? So genomic surveillance is the process of constantly monitoring the pathogens and analyzing their genetic similarities and differences so that we can find out how they are related or different. So if a virus is very much related to Hyderabad and let's say Ahmedabad, so that means this has spread from one city to other and not from let's say Chennai, which is not so much related. So we can actually track the path of the virus and find out how it is spreading. And this is important because we can track, I just said, the spread of the variants, monitor the changes in the genetic code so that we can find new medicines, new ways, and new vaccines even. Collectively, this information helps understand the variants might impact public health, which can help us design strategies to address the problems. Okay, and it is helpful for researchers and epidemiologists, public health officials to understand what is happening. And I will give you some examples how we are now very closely working with the municipal corporations and which is really having real-time impact. So the one weapon that virus and many pathogens have is the mutation rate. They keep changing their genetic structure and therefore the properties, and because they have millions and millions and millions in the number in each patient, so they have a luxury of number. So even if mutation is happening and 99% or more are useless, even then it has a great chance of making new virus and evading the immune system. So the way it happens is that if more and more people are infected, then mean more and more mutation is possible, so that the virus is getting more possibilities. So mutation happen by chance, they are not selected, and they are selected based on the fitness. Advantage will be if it is advantageous, it will be more often. If it is disadvantageous for the virus, it will be wiped out and we will not be able to see it. More mutation doesn't mean more danger, but it just means more possibility in the virus. Some of that can be dangerous. And we have measured that 2 mutations per month is getting established in this virus. So that is the speed with which you are seeing new variants of this virus. So when you want to survey the disease conditions, what we require. So one is clinical samples, that patient is sick, they come to clinic, or workers go and collect their samples, and that is brought to the lab for the processing. So you isolate genomic DNA from there and do the sequencing of large number of samples and look for what kind of changes are happening. The virus is under, by the way, although we have stopped wearing masks or stopped taking so much care, but the virus is still around and there are still large number of cases there. It's just that we have got now sufficient immunity that we are able to handle the virus without going to hospital. Right? So, the selection pressure over the virus is around, it is there, that if there is a new variant that comes which is more infectious, then that will survive because it will spread very fast. The virus which will escape the vaccine breakthrough. So we are all vaccinated now. We have, most of us have taken boosters, those who are of certain age. But now virus will look for how it can break the immune protection that we have. So those will be the variants that are going to come. So the more infectious and more immune escaping variants will be coming in the future. Mortality or worse symptom is of no advantage to the virus. So it doesn't get anything out of that. If it is our bad luck, if along with that higher worse clinical symptoms or mortality comes, but that's not under selection pressure, but it can happen sometimes. So what is the strategy that we are doing? By the way, we are doing very massive scale surveillance right now, as you will see from the presentations, because both the government and many other agencies are interested in following the virus carefully so that we don't get caught in a bad spot in future. So when you want to look at the genome of the virus, you want to look at genome of the virus when there is a more clinical, worse clinical symptom, hospitalized cases. So there can be two reasons. A, either person is himself or herself is weak, so that's why the person is fallen sick, or it is a new variant which is coming, which is more bothersome. That's why any hospitalization should be sequenced, genome surveillance should be done. And vaccine breakthrough. If the vaccine person is vaccinated and got infected, then we should check the virus sequence, because there are two reasons again. One is the vaccine is fading, it's a natural process, or the variant is this new variant which is causing the breakthrough. So that's why we want to explore. And the reinfection, the same thing, where the vaccination reinfection, that is either fading immunity or the new variant. If suddenly you see a lot of cases happening in one part, several hundred people got infected. So there are again two things, either new variant has come or they had partied together in one hall without any mask or something, so that's why they got infected. So later case is not so much to worry, they can win. But if it is a new variant, then we have to worry because this is going to spread in the whole city. Right? So that's why we should quickly find out what is the variant, is a new variant or not. And then of course we should constantly keep checking in different geographical locations. That virus will not evolve only in Hyderabad or Bangalore or Delhi. It will evolve in small places also. So the idea is to keep sampling various parts and keep monitoring if there is a new variant coming or not. And this is not only for this virus, as I will show you later that now we are getting into more and more surveillance. This is just to impress you how we can really follow the virus, different variant, different color is essentially different variant. So this is a timeline. Here they are in April 22 now. The pattern is the same. So you can see here now there are various kinds of this. Here you have one variant, it is replaced by this, then it's replaced by this, then it's replaced by this one. For a long time this Omicron, then this is replaced by some other variant. So as you can see that yellow is more infectious than red, but here the pink is more infectious than yellow, and that's why it is replacing one another. Luckily, so far these have been less and less symptomatic, less and less troublesome. So although we kept getting more and more infection, but there was no hospital problem, unlike second wave when we had lots of issues. Right? So that is how you can monitor what is happening in the city, and you can then pool the data and you can look at it, what is happening in the whole country, and when compile the things. People have now made a system where we deposit the data and monitor world over how these thing changes are happening. So here it shows that weekly number of publicly deposited genomic sequences. As you will see that most of the sequences are deposited from the US or UK largely, and India has contributed very little in this somewhere here, and weekly, now depositing number of cases weekly, even the sequencing. So it's the same cumulative proportion of cases sequenced by the country. We see we are very light color here, so we are very little, and you see the most cases have been sequenced in the only few countries. So that was the case, but even then, because we were, although we were not sequencing that level, but we were sequencing in different places with certain strategy. So you can see here now the earliest detection of variant, because that's the advantage when you detect. There, we are not, this thing is not very bright. So if you see the color of India is something not very light, we are actually comparable to much of the US and Europe. Similarly, this is for the beta variant, this is delta variant. We find well before many other places, you can see, and even the Omicron, we found early on, as the color shows, that along with other countries. So you have to have, it's not the number all the time which is going to be most important, it's the strategy how you collect the samples is going to be very important. So that was the cases that we were collecting samples from patients and then sequencing and following up. Then we switched now a couple of years ago, we started doing environmental surveillance because that was even more effective, and now we believe more and more that that's what is the way to go. You don't have to go to any individual, you just go and collect environmental samples and estimate, technology is there to estimate what is the viral load. You can do calculation, and you can even sequence the samples from the environmental sampling or collections, and even figure out what are the variants present in that. So this, as you would say, is unbiased. It will give the sample of the whole city, and it's convenient and cheap. You have to just, with fraction of cost, you don't have to now sequence thousands and thousands. You have to sequence maybe 5-10 samples. And it's relevant beyond SARS-CoV-2. So when you are doing the survey, you will, any pathogen that is there eventually will end up in the wastewater, and we collect and we know what is the, and that's a big program that we are running in 4-city cluster, where we are following SARS-CoV-2. In addition to that, several other infectious diseases, including AMRs and so on, which are the big problem. So that is the wastewater. I will also discuss, just showcase to you how we do air surveillance. Zoonotic surveillance is another thing which is very important, but I will not have time for that today. So this was very early on, when even before the second wave started, we set up the system to collect the wastewater samples from STPs from several cities. Actually, NEERI is a CSR lab that is spread over, is a National Environmental Engineering Research Lab Institute, which is specializes in sampling, and IICT is another CSR lab, and CCMB put together our efforts and collected samples from different cities and carried out the environmental surveillance. These were one of the first reports. I mean, this is reported in 21, so that was done several months before. At that time, we could very accurately tell in Hyderabad what was the number of people were infected at that time, although the number of cases as from the hospital or individual sampling was about 20 times less than this. So which we knew what level of people infection is prevailing, and we can be better prepared, because most of time cases don't get reported. So now we are not doing STP in Hyderabad, but we are following the open drainage places. So there's about 30 odd open, close to 40 open drainage, and it's funded by various sources. SERB, the DST funding, then Rockefeller Foundation has massive funding for this for 3 years, which is still continuing, and we are able to tell which part of the city, what kind of cases are prevailing. As I told you that we can even do sequencing. So this is the load of the, by PCR method, we can tell that how Omicron is increased over this period of time, and this is the sequencing of the sample, and you can see the red is Omicron, which is increased. So even this just to showcase to you that without going to any individual, we can just go and collect wastewater, and we can tell what is happening in the city with respect to these infections. And these are then, as I said, that we follow around 20 open drainage places, and each individual how the cases are going up and down and so on. So, and this data helps us to understand what is happening in the city, in which part. Somewhere, if there's more cases, mean either new variant is emerging or the people are doing something. It's a human behavior. So this is one example which I thought I'll show you. So this is a small city called Allahabad or Prayagraj now. So you can see these are the cases reported by individual hospital data, and this is the wastewater that we were carrying out. You can see the pattern so nicely matches that whenever there's a drip, you can see exactly the same way. So, but of course the numbers here will be very different. So which means that the actual number of cases are reflected very accurately in the wastewater, in fact more accurately. I'll skip that. These are the same kind of trends that you see. I will spend a couple of minutes, maybe 5 minutes on this aspect. We are doing surveillance in Bangalore in very big way. There are 28 STPs. Bangalore is second largest wastewater system in the world, and we are 80% of that comes in these 28 sewage treatment plants for STP, and we are monitoring all of them on weekly basis with multiple institutions and multiple funding sources. And these are this is map of the Bangalore city, and you can see they are spread all over the place. So here, so I will show you what is the utility of this activity. The red line is the cases reported that you see. This is the third wave that was coming. And the blue is the actual environmental sample that we are taking. So you can see that there have been silent waves that have passed, which was not noted by, you will know if wave is there only if you go and test. If you don't test and people not coming to hospital, we will not know. These were the Dasara festival time when people have interacted more, and we have seen the cases. And these were much later in July this year. Not only do you know the cases, we actually get, depending upon if you do time lag of 7 days or 4 days, depending upon how you do the sampling and analysis, you can predict 8 to 10 days before if the wave is going to come or not. So we keep sharing our data every week with the Bangalore Municipal Corporation, and they know if there is a wave that is going to come or not. For example, there was a lot of stress when the cases started to increase in China, but we were monitoring and we told them that we don't have to worry because it's not happening in India. For a variety of reasons. And this is just, you don't have to read, just to show that these are all 28 different STPs, which represent different wards in the city. And we have exactly granular data for ward-wise, how many cases are there in that part. And so the municipal authorities then take decision that where to focus more attention, where to put more restriction, and where there are no cases, why to put restriction there. People can operate normal life in those places. And this is again from the wastewater sequencing, we can monitor how the profile of variants is changing and which part of the mutation virus is getting mutated more frequently and so on. And then we also do monitoring of various variants, whether they are increasing or not. So you can see BA.2 is more prevalent, while let's say BA, whatever, number doesn't matter, they are less prevalent. And we also discovered, I will not show you those data, we also discovered that this virus is able to recombine. It's not only mutating, it is also recombining like common cold. So this is another tool this virus has, which can accelerate its mutation or variant appearance rates. But so far, we are monitoring many of these. X means this is a recombinant, and we are monitoring many recombinants. And as you can see, I think this is becoming very big. So the recombinants, although they are there, but they are not more infectious or more symptomatic compared to other variants that are around. So it's something to keep in mind, but the virus has that tool for itself. So what we have learned from this is, So our reports, what we published and also generated, is also used in a document that is generated by the World Health Organization about environmental surveillance for the SARS-CoV-2 to complement public health surveillance systems. Right? So because these data that were useful in making those advisories. So what we have learned and what are the way forward is that sewage surveillance is a qualitative and to some degree quantitative estimate of the number of people infected in that population. And this can be used as a reliable and unbiased surveillance measure to understand the progress of the COVID-19 even when the mass scale tests for the individuals are not possible. An indispensable tool for early and easier detection of future COVID and other outbreaks is means to ascertain the efficacy of the strategies in place. You may put lockdown, but if the cases are not coming down, that means your lockdown is not effective, it's only on paper and so on. And then we have given this feedback and which has helped in fixing the problems wherever there was a leakage. And along with SARS-CoV-2, several infectious agents and biomarkers associated with disease can be detected in the wastewater. And wastewater surveillance can help in understanding the antimicrobial emerging in the microorganism. And that's what currently we are doing in many cities. So that was about wastewater. There was a big debate whether this SARS-CoV-2 spreads as aerosol or it is only by touch and something by close contact. So we carried out, so there were a lot of debate about this. So we carried out air surveillance of SARS-CoV-2 and to ask how much the virus can spread through the air. Yes, and that was something very interesting and quite amazing thing what we observed in our study. It's now all published, so you can take a look at some time if you're interested. So we designed a strategy for sample collections. We collected in hospitals, in COVID ward, ICUs, doffing area, corridors, nurse stations, and general wards, mortuary, and so on, to find out where in the air you see the virus. Right? There were lots of problems. The people who die, they were not getting proper handling because people are afraid that somebody died from COVID, so that dead body will be emitting disease and so on. In fact, we reported that in mortuary there is no virus. So unless the person is breathing or exuding something, there is no problem with that and so on. So those are very helpful in for many families for emotional and other reasons. And then we did, in controlled conditions, we made a specific number of people, approach a specific number of people. We made a room where we made them sit for certain hours and collected samples with different air conditions. And even we picked the houses who were willing to be volunteers. And when there are COVID cases, and we tested their bedroom, living room, bathrooms, and so on. So this study was done in 2 cities, in Hyderabad and in Chandigarh, and we have done a lot of samples. It's quite a difficult and challenging thing to. This is our student in the COVID ward near ICU collecting air samples in the ward. So this is how our students they volunteers to go and do these kinds of activities, which were at that time very scary and stressful. And in this, we can find out how many places where the things are viruses around. And this is like, for example, we put the sampler here. Okay, we put the sampler here and then put people for different distance and different time. We dedicated a room for this kind of thing, and they were doing. So COVID positive individuals were asked to spend some time in the closed rooms. Air was sampled a certain distance. Samples were also collected immediately after the departure, and throat swab samples were collected to control that whether there is any relationship with the viral load in the individual and in the air. And the summary is that the SARS-CoV-2 is detected in the air in certain conditions. The chance picking up the virus in the air is directly related to the number of COVID positive cases in the room, their symptomatic status, and the duration of the exposure. the strategy of segregating hospital areas into COVID care and non-COVID care helped in controlling the spread in a major way because the air were cut off from the two regions, otherwise it would have been a bigger disaster. Analysis of air samples collected from houses where individuals had infections suggested that if the patient is immediately isolated after knowing that she is COVID positive, cross infection to other family members can be prevented. So it was we have seen families where infected person is there, but it was isolated in one room and others wearing mask and there were no infection spreading there. So in closed and less ventilated rooms, virus can stay longer, more than 2 hours. We have detected in the toilets where person has gone and used the toilet, and after 2 hours you go and you see the virus in the toilet. So which means when you go to get in the toilet, nobody is there, though you don't remove your mask. This is even more risky that you will have infection there. Or any room where people have been there 1 hour, 2 hours before, don't think it is empty, so therefore you are safe. I mean, this was very, very useful. We made this advisory and it was shared with many organizations and governments to take care of their staff and so on. Right, so these are then the advisories made that if ventilation is there and the people are following certain disciplines, you can very easily avoid large number of infections from happening. So avoid gathering in closed spaces. That was part of our advisory. Avoid spending more time in closed rooms. Keep windows and doors open in the places like classroom and offices or even the houses. And wear mask, especially in a closed space, public toilets, committee rooms, and even if there is no one person there, because somebody may have been there 1 hour or 2 hours before you. So for a lot of this kind of studies, when you put together, many things we have understood, but there will be a lot of debates that will be going on. Some of it is getting heated up also. What is the origin of the virus? What is the variant situation, infectivity, symptom? There will be issues of vaccine equity, because if whole planet is not vaccinated, we are not safe. And then people sharing data with each other. All these issues we have come across in this. And then, of course, long COVID is something which is appearing more and more. We will understand that. But what we can do in the meantime, from what we have understood, is that mask and vaccine as a part of our being considerate to others. It's part of being civil when there is a pandemic of this kind. Avoid clustering and close spaces, which can be done in major to large extent. And remember that virus is spread only through people, not through anybody else. So if we decide, we can block it, but challenging, but possible. In addition to that, I will not be able to show the data, but it's so important. It's not from these kind of studies in otherwise respect nature. If you go and do an excess, it will bounce back on you. All pandemics are originated from animals. And when we go, we destroy their homes, so they will come here, and then we are not used to those pathogens, and some of them pick up and create a bigger problem. All small or big pathogens, largely, which are the pandemic, they come from animals. And of course, the planet belongs to all creatures, so we should not see that all the forest is ours and go and destroy and build agricultural place there. And preparedness for the pandemic in future at global level is very important. One country, one city is not going to help. As you have seen, a variant appears in Africa and within 3 weeks everywhere else. So that's the level. Now going to, I'll just finish in another 2-3 minutes, how we are linking it to One Health. So we have been able to convince municipal authorities wastewater surveillance is effective method for genomic surveillance, and that complements patient data. It can be used to measure infectious disease load, and extremely useful in public health care. In next 2 years, we are going to continue this kind of surveillance beyond COVID, which is other pathogens, including antimicrobial resistance, zoonotic, to have a sustainable system. It's a big thing now, and this is our, so we have 4 city cluster with actually many other small city participants. So like in Bangalore, Hyderabad, Delhi, and Pune are the 4 city cluster, which is very generously funded by Rockefeller Foundation, a program which I am coordinating. But we have other systems, like we have Vijayawada, many other smaller cities are our collection centers, and we work with the local authorities. We collect the sample, estimate, and give first person without any, we have automatic system, without any delay, the data is given to the person or the authority who is given us a sample. So they can make use of it immediately, whether increasing, decreasing. Then we keep doing intercity and national analysis and those kind of things. So now we are working out with what kind of sample collection is done. So manual, or we are also in the process of developing automatic samplers and fit within STPs, and that will collect at a fixed time. So what is the frequency, how much coverage we will do, and transport of the sample, because each place will not have every facility, and then how to store them. Zoonotic sampling is another thing which we are sorting out. So in the water bodies, where we find animal components, insects, because we are not talking only about COVID, we are talking about vector-borne diseases as well, poultry farm, cattle sets, buffer zones where animal, human, wildlife and human conflicts happen, and how to optimize your sampling during the season, like in rainy season, wastewater surveillance is very tricky, temperature, wind, and so on, we are integrating into our analysis. And we are developing strong standard operating procedures so that we can collect the sample, isolate the genetic material, and analyze. If you want to implement at national level, then the training of the people, because each municipality will require maybe half a dozen people to do it, and then indigenization of the components, molecular biology reagents, and automation of sample collection. Some of you may remember that when the COVID began, we were importing all the test kits, and each individual test was costing 4,000 to 5,000 rupees, depending upon which hospital you go. 5,000 rupees. With the government initiative, with the philanthropic funding, within 8 months, we put together more than 100 startup companies, several academic institutions, and we indigenized everything that is required for this, all the way up to units that are used to make the oligonucleotides, which were not made in India. All the raw material was made here, exported, and so on. So we figured out all that, and today you will be surprised to know, pleasantly surprised to know that the production cost of test for COVID test is less than 20 rupees. Where is 4,000, where is 20 rupees? And now we are in the second phase of a large scale production of this thing, and this cost is going to be in single digit. So this is the power of when you optimize and indigenize, then these are actually affordable. So that means in few thousand rupees, we will know the health status of Hyderabad with respect to infectious diseases. So that's the power of science when it comes and integrates with the requirement of the people. And of course, the data integration, what is happening in Hyderabad will have consequence in Chennai very soon, or Bangalore, and so on. So local to city to state to country, and hopefully to neighboring countries, and then integrated through WHO to whole world. There are a lot of efforts going on to integrate environmental surveillance and identify and contain. For example, if there are thoughts that if China was more transparent and came forward much earlier, maybe the dynamics of pandemic would have been different and less painful. But that's something one can keep debating. And of course, this data, real-time data is very important for decision making for the public health. Most of these I have said, so I'm not going to repeat, except that these 3 points, that reporting should be, as I said, that we give the information immediately to the civic authority who gives us the sample. Say they know what to do with their patient or the locality or something. If we see cases are increasing, we tell them this part cases are increasing, they can go and start putting barriers or more restrict mobility and so on. And of course, the accountability and most important public awareness of this. So because pandemics, we also contribute a lot unknowingly. So accurate estimate of pathogen load in the population, eye on the evolution of the pathogen, now even in absence of pandemic, which is not yet finished, but hopefully we are reaching there. AMR and symptoms and infectivity and preparedness for the existing and for the new infection is what becomes possible if we do the surveillance as we are planning in a regular fashion. So I'd like with that to thank all this work was done in CCMB and now TICS is doing, this Tata Institute for Genetics and Society is the main lab now doing the surveillance in Bangalore, and we are doing surveillance in Vijayawada. And very special thank to Bangalore Municipal Corporation and Bangalore Wastewater and Sewage System. They have been so forthcoming and sharing data and even encouraging us to do more surveillance and more pathogens so that they can take better care, while there are other cities where many civic authorities have been very secretive and not sharing the samples because they thought that if number of cases are declared, they will be under some scanner. So that was unfortunate, but many cities are now coming forward, and we are going to set Bangalore as a model how you can implement this and how people get benefited. That's why awareness of public is very important. If you are living in a city and municipality is not doing its responsibility of getting samples properly put for surveillance, they are not doing good job for you. So that's why the awareness is very important. So with that, I'll stop. Thank you very much. I'll be very happy to discuss or answer any questions that you want to.
The session is open for questions. We'll only take 2 questions. If any of the participants are having questions, please ask. We'll take only 2 questions.
Good morning, sir.
Where are you?
Yes. This is Karthik. So my question is, sir, as India has a large population and there are also certain beliefs and many barriers when it comes to collecting data or doing research, how do we tackle these problems?
No, it's a very big challenge. But if we decide to do, we can do it. I mean, if you can implement lockdown, then you can do anything. so it's just question of doing it. It's a large country, lot of population, but if people are aware that we need to keep monitoring our air, our water, we are now setting up, for example, air surveillance continuously in the hospital because there are so many hospital-borne infections that we get, and they are very lethal. So we will know very soon that whether there's a hospital is having enough clean air or not. Are their systems working? So now, so we are now, somehow science has reached a point where we can tell what is the quality of the air here in terms of infectious diseases. Within a day, you could do sampling today, tomorrow you can tell what is there. So hospitals should have better air quality systems so that infections are not, you go there for treatment and come back with infection. Similarly, infections are many times people don't go and tell in the hospital. Like how many dengue cases are in Hyderabad, is in any city or Bangalore for that matter, is always underrepresentation. You will go and you will find there are 100 and so on. But actual cases are more than 1,000 because people don't go to hospital. They take some paracetamol, something, and go for work, because if they go to hospital or a health center, then that day they will not get salary. Most people work on a daily basis. They open shop or they sell something. So that is why reporting is not going to be accurate and will be always under-reporting. Even COVID, we have seen that typically it was 10 to 20-fold less, under-reporting in most cities in the country. So similarly, now, although it is not lethal, it may not, but we know what is the infectious conditions, how many dengue cases. So we can calculate from the water and tell how many people are carrying infection, in which part of the city, how it is changing with the weather. We can match with the vector surveillance, how many mosquito, which mosquito, and those kind of things. So we should realize, because this is very important, if we want to keep our population in better health condition possible, we must know what is the pathogen status. Otherwise, we'll be getting only surprises, and we will doing only firefighting and not the planned proper strategy to take care of that. So this is possible, as I said. So we are doing open drainage, we are doing a small city, let's say Vijayawada, although it's not a small, it's now quite big city. We are doing a very big cities like Pune, Bangalore, Hyderabad, and we are also having plans to set it up in the smaller location like villages where there is no STP, there is hardly any even open drainage system. There are different kinds of things. So we are finding methods how we can, it's like we have a healthcare center all over the country. They should be enabled. If we make proper SOP, make the reagents very cheap, that it just takes few hundred, then they will be doing it and reporting, and we have a very advanced IT system. Is easily possible to do.
Thank you, sir.
Thank you, sir. Thank you, participants. And now, to felicitate our speaker, I would request all the delegates who are gathered here to invite onto the stage. I welcome all the delegate speakers onto the stage.
I can remove.
Yes.
And can I have my? Thank you.
Very important work. Thank you so much for doing that.
Thank you.
Thank you all. Now I request Dr. Scott Hall and Frederick to felicitate our chief guest, Dr. Rakesh Mishra, sir. I would now invite our officials, Sri Hanumant Nayak sir, Additional Secretary T-Sweries, and Sri Jagadishwar Reddy sir, OSD T-Tweries, to felicitate our speakers with the certificates. I request all the speakers to be seated on the stage. I request our officials to kindly acknowledge the international delegates with the certificates. We invite Dr. Amit K. Tiwari. Sir, please present. Certificates. Thank you, sir. Now I invite Dr. Scott Hall. I request Dr. Rakesh Mishra sir to also join in presenting the certificates. Thank you, sir. Now I invite Professor Jairam Puram sir. Thank you, sir. Now I invite Professor P.R. D. Vogel. Now I invite Professor Frederick Williams. Now I invite Professor Hanuman Sagar Bodhu, sir. Thank you, sir. Now I request Dr. Snehal Deshpande to address the gathering for 5 minutes. We welcome you, ma'am.
A very good afternoon to everyone. When I stand here, I can see hearts which are happy and joyful and faces which are bright. I think every single person who has participated in the journey of the last 3 days which we have been together and many months for the organizing team, all of us have changed in every cell of our body. As it is said, neural plasticity is when not only we do something, it is always happening. And it very much depends on the human interactions we have with one another. A beautiful book which I have read is The Neuroscience of Relationships. And I always am inspired by this, that no single brain can grow by itself. We require other brains to grow. Each of us has been endowed with a lot of knowledge, a lot of sharing from our esteemed faculty from India and abroad. I can say from a quote from Harry Potter that we don't need magic to empower or to show the power. Each of us is endowed. We have so much power within ourselves. Go forth and share, my dear ones. I think the success of this conference, the success of all the faculties coming from US here, sharing their knowledge with you, will only reach its point where you go forth and share what you have got with your fellow beings and try to make this world a better place. So, my dear friends, let us join in doing that. Let us make the BioMe 23 a grand success by making the world around us a better place to stay in. Thank you.
Good morning. Now it's the time to announce awards for oral and poster presentation. We'll go for the best oral presentation. The award is, please make it fast to come on to the dais when I announce your name. Mrs. M. Kezia Elizabeth, Best oral presentation. Miss Subhasri Ghosh. So these are the two best oral presentations. And we,
Subhasri Ghosh, is she here? Yes, please. Next, we move on to posters.
okay. Now we move on to posters. Dr. M. Sangeet Kumar, best poster. Miss Shikha Mahati, please make it fast to come on to the dais. Miss Mendelata. Miss Namrata Pal. Miss Shamuda Rahman. Miss Deepti Shika. Macharla Srihari Datta. Miss M. Rakshita. So I congratulate all the winners for their efforts to present the oral and posters. The winners also have gifts from University of Toledo. A big round of applause.
Dr. M. Sangeet Kumar, we request you onto the dais for receiving the best presentation. Sri Haridatta also. I request all the delegates to be seated. Today's students are tomorrow's future, and their contribution will certainly carve the bulwark of success towards the organic growth of the society. The end of the story is the beginning for many others. On this note, let's conclude a remarkable, memorable, and knowledgeable international conference, BioMe 23. I request the organizing secretary of this conference, Dr. Vasanthi, Principal, Telangana Social Welfare Residential Pharmacy College, Mehabad, to please propose a vote of thanks.
Respected all the dignitaries, respected officers of T-Swiss and T-Twist, a very good afternoon to one and all. Words would fall short of describing how much knowledge we could derive from this conference and the important network we could create. This is a momentous conference for all of us. I feel incredible honored, privileged to extend the vote of thanks. I feel overwhelmed by the grand success our conference, which is being initiated by our patron, Sri Ronaldo Sir. Sir helped us in obtaining the necessary administrative approval for organizing the event in Lush Green and Serene campus. Words are not enough to thank Sir for his constant guidance and support to shape the conference. The valuable inputs of Sir make this event a grand success. I sincerely thank Professor Redana Sir, Executive President, Federation of Asian Biotech Association. Thank you, sir, for your enormous advices utilized in sculpturing of this wonderful conference. I express grateful thanks, Mr. Shakti Nagappan, CEO, Telangana Life Sciences, for providing opportunity to visit pharma companies at Genome Valley. I am very much thankful to all the sponsors, Government of Telangana, DST SERB, and TSCHE. Without their generous financial support, it would not have been possible to organize this conference. I would also like to thank Telangana Life Sciences, FABA Academy, and Heartfulness Education Trust. Wholehearted support has made this conference a reality. I acknowledge the unwavering support received from officers of T-Swiss and T-Twist, which led to the success of this conference. We look forward for your support for all our upcoming programs. I thank all distinguished delegates who have accepted our invitation and giving generously of their time. Your participation added depth to the content and deliberations. Thank you for coming. As event of this magnitude requires meticulous planning and execution. My heartfelt thanks to Dr. A.S. Pavani Ma'am, OSD, HE, T-Swiss, and convener of BioMe. Ma'am initiation and involvement at every step made the task easier. Thank you, ma'am. I would also express thanks to Dr. Jagadish Gandla, COO, FABA Academy, and co-convener of BioMe for his endless support. I can't thank everyone by name, but I must place on record appreciation of the untiring efforts by organizing committee. I would like to thank press and media for splendid coverage of the day-to-day event. I am thankful to all the participants for your active participation in this BioMe. Thank you each and everyone for joining us and making this conference a grand success. Thank you one and all. Jai Hind.
This is regarding a special wholehearted gratitude to Dr. A.S. Pavani, OST Higher Education Wing, T-Sweries, for being with us right from the time the seed of this conference was sown till now when it stands before us in the form of an enchanting tree. Ma'am has been glorifying this journey just like a sun paving the way to this success today. We thank you so much, ma'am, for your incredible support and for playing a key role in the success of the conference. Thank you each and everyone.
I request everyone to stand for national anthem. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभाशिष मागे गाहे तव जय गादा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जय हिंद. Thank you. I quickly call the organizing committee members for a picture, for a group picture, and the officials, organizing committees, officials of T-Sweries and T-Tweries. I request all the organizing committee members, officials of T-Tweries and T-Sweries for a group picture with the speakers. I request you all to gather quickly. I request the participants to have lunch in front of in the dining hall, in front of studio rooms, in front of student dorms.